Okay. So, cysteine desulfhydrase. All right. Experiment number 26. So, cysteine is just an amino acid. So we talked about amino acids with regard to having a carboxylic acid group, an amine group, and then that R group coming off, and it has a hydrogen right there. So all that stuff applies. It's just it's just one of the amino acids that contains a sulfhydryl group. What a sulfhydryl group is just a SH. A lot of y'all do with like alcohol groups, right? So OH. S is like right underneath the O on the periodic table, so it has a lot of similar properties that oxygen has. So SH, OH, they're very similar, right? So and what's special about sulfhydryl groups is that they can form disulfide bridges. All right. So hold on, let's take a look at this real quick. So this is cysteine. This is the amino acid cysteine right here. So I'm not lying to you, right? It's got the SH right there. Got the carboxylic acid. Got the amine group, right? And then the SH right here. And it's got a carbon right there too. So what a disulfide bridge is when you take two cysteines, so disulfide, right? So two cysteines with two sulfurs, and you form a bridge with them. You connect them. So you take away the two H's right here, and you connect them right here. So this is a disulfide bridge like that. All right, so it's pretty cool. And it's very important for like a lot of stuff. So what cysteine desulfhydrase is that's an enzyme that can break cysteine down for energy. All right, so cysteine desulfhydrase is the first enzyme of the breakdown of cysteine, all right? And so what it does is, this is the reaction that occurs right here with cysteine desulfhydrase. It's H2O plus a cysteine yields pyruvate right here, H2S, which is a lot like water, just with sulfur instead, and ammonia, so it takes off that NH2 as well. All right, this pyruvate right here, if you remember, right, glucose is broken down into pyruvate, right? And this is broken down into pyruvate. So this pyruvate can then enter the mitochondria, can then be oxidized, and then enter the Krebs cycle, and all that occurs. Or it can be fermented, right? Or no, it can't. No, it can't. Never mind. Back that up. Because you need glycolysis to do fermented, right? So pyruvate right here can be used to break it down as an energy source, right? As pyruvate. So what we did was, we took peptone iron agar, is an agar that we use, and it has a lot of proteins, right? So it has a lot of proteins, and amino acids make up those proteins, so it has a lot of cysteine in it, all right? And it's also got a chemical called ferric citric in it, which we'll talk about later. So what we did was we took a deep of peptone iron agar like this, and we stabbed it with a needle, right, that had microbes on it, and the microbes have the enzyme cysteine desulfhydrase. So because it has cysteine desulfhydrase, it undergoes this reaction right here, right? And it produces a lot of H2S, right? Dihydrogen sulfide. Or hydrogen, no. It's just hydrogen sulfide, right? So hydrogen sulfide is a gas, okay, and it's soluble. So it's gonna be soluble in this peptone iron agar. And so the reason why we have ferric citric is because ferric citrate then reacts with hydrogen sulfide to form ferric sulfide, FeS. And that right there is a black precipitate. So if you stab the deep in with microbes that have cysteine desulfhydrase, then you'll produce this black precipitate like you see here, okay? And if it doesn't have cysteine desulfhydrase, you stab it and no blackness is going to occur. Right? If you kind of look really, really closely, it's kind of, you can see like the stab, but there's no black in it, right? Forgot to put the results in, right? So your microbe that we used is Citrobacter frundi and Escherichia coli. Remember, E. coli is your bitch bacteria, so that doesn't have a black stab line in it, while your Citrobacter frundi will have cysteine desulfhydrase. So that's going to be your results here. So the big takeaway that I didn't put in is you just have to know that amino acids can be broken down, right? So we demonstrated cysteine desulfhydrase as like our model example, but a lot of these amino acids can be broken down and used as energy. It's the reason why it can be used as energy is because they're metabolic intermediates. So something like pyruvate is in the middle of your metabolism, and so you can use that pyruvate as energy, right? But it's a relatively straightforward and simple experiment. You're just looking for that black stab line like that. All right.